Hey guys, so I hope you had a good weekend and we are back in class. Okay, so we're going to pick up where we left off last time. So we've been talking about these glycoproteins, right, which are basically protein mixed with a carb. And last time we talked about these O-linked saccharides, as we call them. Now we're going to talk about the N-linked saccharides. Okay, so last week on Friday we were talking about how the O-linked saccharides or when we link carb groups to polypeptide chains via the hydroxyl groups of serine, threonine, or a sort of not pure amino acid, but a version of an amino acid, hydroxylysine, where we've hydroxylated along the backbone. And I showed you a picture of this back when we talked about amino acids before. But now we're going to focus on N-linked saccharides, okay? And how we do our N-linked saccharides is it's always through the amide nitrogen of asparagine. Okay, so here we have asparagine. Here's our sort of main group structure. It's always going to be on the R group, right? Because the main group of your amino acid is going to be in the middle of a chain, right? And the asparagine doesn't have to be like C terminus or N terminus. It can be at any point on this peptide chain, which means it needs to be the R group, right? But it's this amide group that can then attach to these sugars, okay? So let's look at some examples. So there are three primary types of N-linked glycoproteins that we see. One of them, and it's my favorite just because of the name. I mean, how cool is that? It's called high mannose. It just sounds like so official. High mannose. High mannose is literally, it's just a whole bunch of branched mannose. Okay, like a whole bunch of branched mannose. We have complex glycoproteins in which you actually see sort of you'll see branching, and the chains are just a big variety of all different types of sugars, right? So just a lot of variety in what you're seeing. And then we have hybrids. And hybrids are, there's going to be one branch chain that's just mannose, so similar to high mannose, and then you're going to have a branch chain that's more similar to the complex. So it's literally a hybrid of these two. But these are pretty much what we see as far as the N-linked glycoproteins are concerned. Now, but we need to talk about like why we care about them. Okay, so these N-linked glycoproteins, these N-linked oligosaccharides, they alter, and honestly, it's not just if it's N-linked, it's anything. Anytime you change or you attach something to a protein, any sort of a post-translational modification, you're going to change the chemical and physical properties of that protein, right? You're going to change its solubility. You've made it heavier, right? You could have affected its electrical charge depending upon what you attach to it, right? And these carbohydrate moieties, which basically means functional groups, right? Think of this as these carb residues. That's another way to say it. They're able to stabilize these protein conformations because they act as sort of like an anchor, right? Think of a ship with an anchor. It drops anchor in the ocean to sort of stabilize it. These carbs can kind of act like anchors for these, and they can help protect the proteins from being broken down by various enzymes. So if a protein is incredibly important and needs to get from point A to point B before it's broken down, if it has the ability to sort of form this N-link oligosaccharide, it's going to do that because it helps protect it. And so we are eukaryotic organisms, right? They like these post-translational additions of these N-linked oligosaccharides because they can help get proteins from A to B, especially when B is a membrane compartment. And another reason why we like looking at this N linkage is because this can actually serve as almost like a timing device for degradation, because basically what happens is you can, so in this example, what we're going to do is we're going to start cleaving off this sialic acid, right? And whenever we do this, it exposes this galactose residue. And we can, so the sialic acid goes away, and now we have this exposed galactose residue. Well, we can keep doing this. We can keep releasing the sialic acid, exposing a second galactose residue, pop off the other sialic acid, exposing a third galactose residue. And the more exposed galactose residues you have, the more likely this molecule is to bind, especially in the liver. okay? So cleavage, this is sort of a cleavage process where we're specifically looking at in blood, where we're targeting for degradation in the liver, okay? So the end linkage just really helps again by anchoring and allowing us to do this sort of time-wise degradation where we can actually link better. Because if you need to do releases one at a time, having an anchor is going to help you with that. Okay? All right. So we've talked about glycoproteins. We know that a glycoprotein is a protein carb hybrid. It's a protein with carb on it. So then what is a proteoglycan? Because that's sort of the next big thing we need to talk about here. And what a proteoglycan is it is a glycoprotein, right? But it's a glycoprotein whose carb functional group, right, whose carb residues are predominantly one thing. They're predominantly glycosaminoglycans that are O-linked to the serine residues. Now, we've already talked about sort of the 6 primary ones of the glycosamino. We're having an issue here, and PowerPoint is freezing. Great, lovely. Sorry about this. Not really sure what's happening. Hopefully we can get out of it. Oh, great. All right, I'm just gonna pause the video. We'll come back. And we're back. Sorry about that. Apparently, my computer decided to go all spaz attack on me. All right, what were those glycosaminoglycans? I was literally just trying to show you this slide again, right? These were the ones that we've already talked about, right? Heparin, which helps with blood clotting. We'll be talking about that some more. We'll definitely be talking about this chondroitin sulfate. So we have a 4-sulfate and a 6-sulfate where it's exactly the same sugars involved. So it's the same disaccharide. It's just a different linkage, right? Linking at the 4-carbon versus the 6-carbon. We're going to be talking about all of these. Hyaluronate, okay? So you may want to sort of flip back to this as we're talking about these to recognize what sugars exactly we're talking about. But let's go back to, here we go. All right, so proteoglycans, right? We're talking about glycoproteins whose carb groups, whose carb residues are basically what I just showed you, right? Those glycosaminos, glycans, all right? And then they're going to be O-linked to serine residues, okay? Now, these things range in size from being small, attached to small proteins, to attached to huge, 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 huge proteins. These can be soluble proteins, or these can be insoluble proteins that are transmembrane, right? If it's in the middle of a membrane, that's going to be insoluble, all right? So, and you know, these are just some examples. Decarin is an example. I've got pictures on the next slide. It doesn't have a lot of linkages here. It's only got 3 options to sort of attach carbs to. Whereas, look at this one, this cartilage matrix proteoglycan, which I've got a video for you guys to watch on how cartilage works. This has a whole bunch of serine glycine pairs in which these chondroitin sulfates can attach to, right? Which is important because it helps us with hydration in our joints, okay? Because there's this fluid in our joints, and if the fluid's not in your joints, that's not going to end very well, right? So we need that fluid in our joints. Here's some pictures from your book, right? So this is an example of a proteoglycan, and you see that every single one of these are the little sugar functional groups. So these are these disaccharides coming off, okay? So this is chondroitin sulfate. It's not specifying what the linkage is here. Don't worry about that too much. And this is the protein core, right? So here's your C-terminus, and here's your N-terminus, and it tells you that this part is really important. It's a binding domain for hyaluronic acid. Okay, here's another one in which, oh dear God, look at all these. I mean, like there's literally no space in between these different sugar functional groups, all right? And then look at decarin. Now, it could make 3, but we only see 1 here in this picture. Here's one that shows you that this is a transmembrane protein. So you've got this portion of the protein is going to be your nonpolar residues, and then polar residues down here. Here's your C-terminus and N-terminus. And in this case, we've got a couple of different types of sort of these glycosamino proteins attached to it, right? Or glycosamino sugars, right? These disaccharides. And then here's another one. So we have a huge, much, much, much larger, lots of hydrogen sulfates all over it. You've got various different O linkages. You have keratin sulfates attached to it. You have N-linked oligosaccharides. So you can get some very interesting stuff as far as these structures are concerned. They can be very complex or very simple. Okay, so what's sort of the function of these proteoglycans? Well, they serve a variety of functions, right? But for the most part, they make up these extracellular matrices, which is really important. And they are involved in a lot of our binding sites, okay? So many of these are involved in the binding sites of proteins that they attach to. There are growth factors associated with them, so binding sites and growth factors, and they do make up a large portion of extracellular matrix, which in the video I'm going to show you how it serves a role in working with cartilage in our joints. So these proteoglycans can actually help modulate cell growth. Like for example, heparin and heparin sulfate, which I spelled this one wrong, my bad, inhibit sort of cell proliferation. So, and how they do this is there's this internalization of the glycosaminoglycan sort of functional group, and it migrates to the cell nucleus. So how it's able to do this is heparin, right? Which we can see in the structure down here. Heparin is a carb that has anticoagulation properties, right? It's really important. And this sequence that you're seeing right here, this sulfated pentasaccharide sequence, it binds to antithrombin 3, which allows our blood to help start coagulating. And so it's very important, and it is able to migrate very well throughout the body and perform these functions wherever it needs to. So it can prevent things from taking place, and it easily moves and migrates to get to where it needs to get to. So let's talk about some other things. Well, fibroblasts, right? The growth factor that tightly binds heparin and other glycosaminoglycans. This protects. So this growth factor protects the growth factor from degradation enzymes, from enhancing it. So basically what it means is we can sort of build up a whole bunch of growth factors for cells to use later on. Imagine it is just like a pool for, it's just so, these things are so good at binding other things, so it can bind all these various growth factors to itself and then hold them until we're ready to use them. It can also transform some of these growth factors, and that can stimulate synthesis and secretion of more proteoglycans. So not only can it store these growth factors, but it can actually transform them and use them for its own purpose. And several proteoglycan core proteins have domains that are very similar to those of the epidermal growth factor and complementary regulatory factor. What I'm trying to say here is don't get too mixed up with the words. Just the big story of these are not performing sort of minor tasks that don't matter. Like your epidermal growth factor and your complementary regulatory factor, these are huge, huge things. And these proteoglycan core proteins have domains very similar to this. So this drives home, again, their importance and how we need these things. And this is an example of how proteoglycans make cartilage resilient. Okay, so in cartilage, these long fibers, right? Long filaments of hyaluronic acid are studded and coated with all these proteoglycan molecules, so all the little blue points are these proteoglycan molecules. And basically what this does is it helps to keep the joints hydrated. So when a joint is sort of not compressed, there's a lot of water and fluid in there, and then when you bend your knee and you compress that joint, the water is able to squish out. Basically, it allows for almost like a squishiness effect by this structure. All right, so we'll come back in the next video and keep talking about cartilage a little bit more.